Welcome to the Bio Insights, where science comes alive.
I am absolutely thrilled you're here today because honestly, we are looking at a topic that is just, well, it's nothing short of mind-blowing. We're going to bridge this incredibly captivating mid-century historical mystery with the modern engineered world that you and I interact with every single day. We are exploring the story of biotechnology. It's really a journey from understanding the very basic building blocks of our existence to literally learning how to rewrite them. So think about this for a second. What do a fluffy loaf of bread, a life-saving synthetic vaccine, and the fundamental mystery of life actually have in common? I know, it kind of sounds like a riddle, right? But the answer connects the oldest human traditions with the absolute cutting edge of modern science. The answer is the manipulation of living processes, and that all boils down to the code that instructs life itself. Section 1. The Mystery of Life. The Race to Uncover DNA. Okay, let's dive into this. To understand how we engineer life today, we really have to go back to a time when life itself was a complete physical mystery. I mean, by the 1940s, scientists knew chromosomes existed in cells, right? And they knew they were made of protein and this other stuff called deoxyribonucleic acid, or DNA, but nobody knew its shape. Nobody knew how it actually worked. Then, in 1944, the famous physicist Erwin Schrödinger published a book called What is Life? And he essentially threw down a massive gauntlet. How does a physical gene encode the information of life? Well, the race was on. In 1951, American chemist Linus Pauling used X-ray crystallography to show how proteins fold into spirals. He guessed DNA was a triple helix, which, actually, scratch that, it was wrong, but he was getting close. Then, in 1952, Rosalind Franklin used that incredibly finicky X-ray technique to capture an image that changed everything. And finally, in 1953, James Watson and Francis Crick put the pieces together. Section 2. The Alphabet of Biology. Decoding the Double Helix. Now, identifying that DNA was the physical material of life was one thing, but actually figuring out its shape and decoding its alphabet, that was the breakthrough that opened up the entire field of modern biology. Now, what's really interesting about this dynamic is the stark contrast in how history treated the people involved. On one side, we have Watson and Crick. They built the famous 3D models out of tin and cardboard, achieved massive scientific celebrity, and shared the 1962 Nobel Prize. But on the other side, we have Rosalind Franklin. Her X-ray crystallography work, specifically that famous Photo 51, showed a clear X pattern, which is the absolute signature of a helix shape. Without her knowledge, her deputy director secretly showed this image to Watson and Crick. They relied heavily on her data to realize DNA must be a double helix. Tragically, Franklin died prematurely of cancer, likely from her X-ray work, and because the Nobel isn't awarded posthumously, she was entirely uncredited during that famous 1962 ceremony. It wasn't until historians dug into the story much later that she was rightfully placed as a protagonist in the discovery of DNA. So once that double helix structure was understood, scientists realized something incredible. Life itself could theoretically be read and programmed. Think of it exactly like computer software. DNA has 4 nucleic acid letters, A, T, G, and C. It acts as a machine language program. That code is transcribed into RNA, which is kind of like the assembly language. And that RNA instructs the creation of proteins, the actual physical building blocks of everything alive. This step-by-step biological flow is what Francis Crick called the central dogma of genetics. The beautiful zipper shape of the DNA double helix allows it to just split right down the middle. Because an A base always pairs with a T, and a C always pairs with a G, a cellular machine can literally just look at one half of the unzipped staircase and perfectly rebuild the other half. It's a biological copying mechanism with very, very few errors. And when rare errors do happen, well, those are mutations, the very molecular basis for evolution. Let's move to Section 3, Defining Biotechnology, Life as a Technology. Once you understand that biology operates on a programmable genetic code, a massive paradigm shift occurs. We transition from merely studying biology to actively engineering it. So, what exactly is biotechnology? Simply put, it's the utilization of living organisms, their parts or biological processes, to develop products or processes that improve human life. Now, in a classical sense, this isn't totally new. If you've ever baked a loaf of bread using yeast or enjoyed a glass of wine, you've engaged in classical biotechnology, for sure. You're using a living microorganism, yeast, to run a fermentation process. But modern biotechnology? Wow, that is a whole different ballgame. The transition from just baking bread to modern genetic engineering happened right here in the early 1970s. Scientists realized that just knowing what genes code for what traits wasn't enough. They needed a way to move those genes around. Enter recombinant DNA, or rDNA, a real game changer. Researchers like Paul Berg, Stanley Cohen, and Herbert Boyer successfully cloned sections of DNA from one organism and spliced them into the genome of a totally different microbe. They literally cut and pasted the code of life. This turned abstract scientific theory into an applied, world-changing technology. Section 4. Biotech in Medicine. Healing the World. That world-changing technology hit the medical field like a freight train, birthing big pharma and a completely new standard of healthy living. These applications alone have saved countless lives. Before recombinant DNA, people with diabetes had to rely on insulin extracted from the pancreases of pigs and other animals. But by splicing the human gene for insulin into bacteria, scientists could grow vast vats of pure synthetic human insulin. Then consider the hepatitis B vaccine. It used to be derived directly from the blood of infected patients, which, as you can imagine, was a painful and risky process. But biotech allowed us to synthetically produce a recombinant version, using just a small, totally safe part of the virus. It's so effective that it drastically reduces the chances of liver cancer, literally making it the first anti-cancer vaccine. Add in monoclonal antibodies that differentiate cancer cells from normal cells, and gene therapies that directly target DNA, and you can see how medicine has been completely revolutionized. Section 5. Biotech in Agriculture, Feeding the World. But this technology didn't just stay in the pharmacy. Oh no, it moved directly into the fields and onto our dinner plates to solve specific, real-world agricultural problems. So the crucial point here is that the science is sitting right in our grocery carts. See lactose-free milk on your grocery store shelf? That's biological processing at work for people with intolerances. But it goes way deeper with genetically modified organisms, or GMOs. Take BT cotton. It is engineered at the DNA level to be naturally resistant to pests, which means higher yields for farmers and way less chemical pesticides sprayed into the environment. And perhaps most incredibly, look at golden rice. This rice was genetically modified to appear yellow because it's packed with beta-carotene, which the human body naturally converts into vitamin A. Since rice is a staple diet in many developing nations, golden rice was specifically designed to combat severe malnutrition and prevent vision loss in vulnerable populations. We are literally engineering crops to be self-contained vitamins. How cool is that? Section 6. Our bioengineered future, industry, and the environment. Medicine and agriculture are just the beginning, really. As we push further into the 21st century, biotechnology is completely reshaping our heavy industries and giving us brand new tools to protect the environment. The scale of this is absolutely staggering. At the macro level, we have bioremediation. We are literally designing and deploying microbes whose biological function is to eat up oil spills and detoxify polluted environments. We're also engineering organic materials into biofuels to offer real alternatives to fossil fuels entirely. Then we zoom all the way in to the microscopic level. Nanobiotechnology allows us to manipulate single molecules for ultra-small, perfectly timed drug delivery right inside the human body. And to understand all this massive amount of data, well, we use microarrays, tools that allow scientists to monitor the activity of tens of thousands of genes simultaneously. It is biology perfectly married to advanced microelectronics. It's like a giant puzzle coming together. But as you might guess, all this incredible science inevitably collided with business and the law. In 1980, a massive legal landmark was set in the U.S. Supreme Court case Diamond v. Chakrabarty. The question at hand was, could a company patent a microbe that had been engineered to eat oil? The court ruled yes. They decided that if you purposefully engineer an organism's genome, you have transformed it from a natural phenomenon into a patentable technology. This ruling changed the world overnight. Life itself became a potential technology. It triggered an absolute explosion in the biotech industry, leading to massive initial public offerings. Universities suddenly realized their biology departments were sitting on billion-dollar technology transfers. The whole shebang shifted. Which brings us to a really profound stopping point for today's explainer. We've journeyed from Watson, Crick, and Franklin unlocking the mystery of the double helix to an era where we can program DNA like software to cure diseases, feed nations, and clean the oceans. But I want you to really think about this. As scientific knowledge turns the very building blocks of life into patentable, programmable technology, where do we draw the line? It's a question that blends science, ethics, and our very future as a species, and it's one we will all have to answer together. Thank you so much for exploring this with me today, and keep asking those big questions.